Okay, so hello everyone. My name is Hala Mahmoud. I'm a second year medical student. First of all, Ramadan Mubarak. I hope everyone's doing well. So today's PAL is going to be regarding the metabolism of amino acids. Now, previously, it used to be given as two separate lectures, but they decided to combine it this year as one. For the first part will be about the products that we can get from amino acids. And the second part will be the inborn errors of metabolism. So first we'll talk about the things we get from tryptophan, histidine, glycine, arginine, and glutamate. And then we'll mention a few diseases related to the inborn errors of metabolism. So Bismillah, let's start. First, we're going to start with the products of tryptophan. So tryptophan is an amino acid that can give us three different products: serotonin, niacin, and melatonin. So let's start with serotonin. How is serotonin produced? So first of all, tryptophan is hydroxylated, meaning you add a hydroxyl group, and it's converted into 5-hydroxytryptophan. And then 5-hydroxytryptophan is converted into 5-hydroxytryptamine. So 5-hydroxytryptamine is basically the same thing as saying serotonin. It's just a different name. And for this amino acid, I don't think you need to know it. Just focus on this one. This is the rate-limiting reaction. And the enzyme here, this applies for almost all of the cases. The enzyme always makes sense in terms of its name. So tryptophan hydroxylase, it's a hydroxylase enzyme, meaning it adds a hydroxyl group. And because it acts on tryptophan, so it's a tryptophan hydroxylase. And the cofactor that is needed for this enzyme to function is BH4 or tetrahydrobiopterin. Okay, so you need this cofactor in order for the enzyme to work properly. Okay, so tryptophan, 5-hydroxytryptophan, and then serotonin. Like, what is this cofactor that I just mentioned? What is tetrahydrobiopterin? So first of all, if you break it down, tetra for 4, hydro for the hydrogen, and then biopterin. Where does it come from? It comes from dihydrobiopterin. So if you take dihydrobiopterin and you add 2 hydrogens to it, that makes sense, right? Here it's 2, it becomes 4, so you need to add 2 more. So as you can see here, you add 2 hydrogens and then you will convert it from dihydrobiopterin into tetrahydrobiopterin. And the addition of a hydrogen is also known as a reduction reaction. That's why we call it a dihydropterin reductase. Again, the enzyme does reduction, so it's a reductase enzyme, and it acts on this, so we call it dihydropteridine reductase. So that's where we get BH4 from. So if we go back again here, this is where we got the cofactor from, and we need it for this enzyme, tryptophan hydroxylase, to work. Okay, but where does it get this hydrogen from? We said that it adds 2 hydrogens from to convert it from dihydropterin into tetrahydrobiopterin. It gets this hydrogen from the NADH. So it takes the hydrogen from here, and you're left with NAD+. Okay, but we just mentioned how serotonin is produced. What happens after we produce it and release it? And basically it binds to the receptor and it does all the effects that it's supposed to do. What happens after that? The serotonin will be reabsorbed or it will undergo, basically it will enter the transporters again for reuptake, and then it will either be broken down or stored into vesicles. So if it's broken down, that's going to be done by the MAO enzyme, monoamine oxidase, or it's going to be restored into vesicles to be recycled, because our body loves to recycle materials instead of just synthesizing it again and again and again. So two things. If we're looking at the fate of serotonin, it's either going to be broken down by MAO or stored again. And it has to be reabsorbed by this reuptake transporter. طيب? So in some people, they have a disease known as depression. When you have depression, that means that you have low dopamine and low serotonin. طيب? So low dopamine and low serotonin. So a way to treat that is for you to inhibit one of these two pathways. So first, we'll talk about MAO inhibitors. What MAO inhibitors do is that they inhibit this enzyme in the mitochondria. So you're not going to be able to break down serotonin anymore. So when you don't break down serotonin, you have higher levels of serotonin available. Remember, we said that the problem in depression was low levels. So we're trying to bring it back up. So MAO inhibitors will inhibit this breakdown. You'll have more serotonin available, and that's good. It actually works on both serotonin and dopamine. Now for SSRIs, or selective serotonin reuptake inhibitors, what they do is that they block this reuptake transporter. So you will not be able to reabsorb the serotonin and take it back into the neuron. So it's going to stay here in the synapse. So more available serotonin here, so it's going to bind more and do the actions that it's supposed to do. Okay? And can you guess which one is more strong or which one is more potent? It's going to be the MAO inhibitor. And that's because we said that MAO inhibitors work on serotonin and dopamine, and it even works on other neurotransmitters as well. But SSRIs work only on serotonin. It's selective for serotonin. So this is more potent. Okay. طيب. So just a recap, like in case the drawing wasn't very clear. When we have depression, we have low dopamine and serotonin. In order to treat that, we can use a type of antidepressant known as a MAO inhibitor. It inhibits the MAO enzyme. So we won't have breakdown of serotonin and dopamine. So you will have higher levels of serotonin and dopamine in the brain, and it's more potent. So this is the inhibitor binding to the MAO enzyme. And then the other antidepressant is selective serotonin reuptake inhibitors. What they do is that they inhibit the reuptake. So you don't have reuptake or reabsorption of serotonin. You block this transporter here. So we have more serotonin available, and you have more of the effects of serotonin. Okay, so we talked about how tryptophan synthesized serotonin. The second thing we're going to discuss is melatonin. So melatonin is a hormone that is produced and released by this gland here, known as the pineal gland. And this gland is important for regulating your sleep. So at night, when it's dark, this gland starts to release melatonin in order to regulate your circadian rhythm and your sleep-wake cycle. Type. This hormone, melatonin, comes from serotonin. So after tryptophan gives us 5-hydroxytryptamine, and then 5-hydroxytryptamine, tryptophan, and then 5-hydroxytryptamine, it will give us melatonin, and this is the hormone that is going to help us in the sleep regulation. I just want to note, try not to confuse melatonin with melanin. So like melanin is the pigment in our skin, in our hair, in our eyes, but melatonin is the hormone that helps in sleep. The third thing that we get from tryptophan is niacin. So I realized that you guys took vitamins in MOL 1, so you probably already know that niacin is vitamin B3. But just in case, this is something that helped me remember it. It's kind of stupid, but you don't have to use it. But when you say niacin, so it's like niacin. Sin sounds like letter sin in Arabic. So if you flip it around, it looks like a 3. That's how you remember that niacin is vitamin B3. You can completely ignore that, but that just helps. Anyways, tryptophan gives you niacin or vitamin B3, or you can even call it nicotinic acid. And then the niacin gives us niacinamide, which is also called nicotinamide. Nicotinamide is an important component of NADH and NADPH. You can even see it in the name, nicotinamide adenine dinucleotide. So tryptophan gives us vitamin B3 niacin, which is going to also give us NADH and NADPH. And one important thing is in this part right here, the red dot, we need an important cofactor, which is vitamin B6. So remember, you need B6 to make B3. Okay, so you need vitamin B6 to make vitamin B3. Right. So now let's kind of try to review everything we just mentioned about tryptophan. Tryptophan is converted to serotonin. And what is the intermediate that we get before serotonin? So I'm just going to give you like 5 seconds to think of the answer, and then I'm just going to reveal them. So tryptophan gives us 5-hydroxytryptophan, and then gives us 5-hydroxytryptamine, the other name for serotonin. But serotonin synthesis requires, I should have covered this. It requires which enzyme? Tetrahydrobiopterin. No, it requires which cofactor? Sorry, tetrahydrobiopterin. And which enzyme? Tryptophan hydroxylase, because it hydroxylates the tryptophan amino acid. So again, tetrahydrobiopterin. What gives us this tetrahydrobiopterin? Where do we get it from? The enzyme is DHPR, so dihydropteridine reductase, which gets the hydrogen from NADH. So dihydropteridine reductase takes the hydrogen from here, combines it with the dihydropteridine to give us BH4. Okay, now for serotonin degradation. We said that for people who have a certain illness, they use monoamine oxidase inhibitors. So what type of drug is this? Antidepressants. So you treat depression by preventing what? So what does MAO inhibitor do? What does a MAO inhibitor do? It prevents the breakdown of both dopamine and serotonin. So is it more potent or is it less potent than SSRIs? More potent because they affect more neurotransmitters. Remember, MAO inhibitors will affect both dopamine and serotonin, while the SSRIs will only affect serotonin. Now, tryptophan also gives us niacin. What's another name for niacin? Vitamin B3. What is the cofactor that we need to convert to get B3? B6. Remember, you need B6 to make B3. And then from niacin, we can produce two things, NADH, NADPH. Okay, so that's it for tryptophan. Now for histidine. Okay. Now for histidine. Histidine is an amino acid that can give us histamine. So this one's pretty simple because they sound very similar. Right? So histidine gives us histamine with the enzyme histidine decarboxylase. So again, let's try to make sense of the enzyme. This is a carboxyl group, COOH. When we remove this carboxyl group, we get histamine. So it's a decarboxylase that removes the carboxyl group. And it acts on histidine, so histidine decarboxylase. And again, it needs vitamin B6 as a cofactor. So far we have tryptophan hydroxylase and histidine decarboxylase, both needing niacin production needs B6 and histamine production needs B6. Okay, I'm sorry about that. Tryptophan hydroxylase does not need B6, it needs tetrahydrobiopterin. Okay. Now we're done with histidine, we're done with tryptophan, we have glycine. Glycine gives us heme and creatine. So let's start with heme. So we all know that our red blood cells have hemoglobin, and hemoglobin helps us carry oxygen throughout the body. So what makes up hemoglobin? Hemoglobin is made up of heme and the globin protein. But now we're going to focus on heme. What is heme exactly? Heme is basically a porphyrin ring plus iron in the middle. But we want to know how we produce this heme. Where does it come from? It actually comes from glycine and succinyl-CoA. So when these two components combine, they give us ALA, and then you don't need to know this part. It ends up combining with iron to give us heme. So this is kind of another stupid mnemonic. You can ignore it, but I'm just trying to think of ways to make it easier to memorize. So we said glycine with succinyl-CoA give us heme. When you think of heme, you think of blood, right? And when you think of blood, you think of vampires. So vampires sucking blood, and it's a gory scene. A gory scene, like a scary, violent scene. So a gory scene of vampires sucking blood, think of glycine, succinyl-CoA, and heme. Okay. Now, glycine also gives us creatine. So we mentioned heme now. Let's talk about creatine, which needs both of these amino acids. Okay. How is creatine synthesized? We have three components: glycine, arginine, and a methyl group from SAM. So we're going to mention SAM again later on, but basically it is a methyl donor. It gives up its methyl group. So glycine, arginine, and a methyl group. These three combine to give us creatine, and then creatine is phosphorylated. It takes a phosphate group from a phosphate donor. This time it's ATP. So ATP gives up its phosphate to become ADP, and then they create creatine phosphate. Right? So when you phosphorylate creatine, it becomes creatine phosphate, or you can also say phosphocreatine. Like, why is this phosphocreatine important? It's basically a source of energy or a source of ATP in your muscles. Like, you might wonder, why do we produce a source of ATP when we could just have ATP? Like, why not just store ATP? This is because phosphocreatine is a bit more stable, so it might be more available. And whenever you need energy, you can quickly break down phosphocreatine to give you the phosphate to produce ATP back again. Okay, so phosphocreatine provides a phosphate group that can be donated anytime you need it to ATP. Type. Now, another mnemonic for this is if you remember that creatine gives ATP to muscles. So GAM, G for glycine, A for arginine, and M for methyl group from SAM. Okay, so creatine gives ATP to muscles, glycine, arginine, methyl group. Okay. So this is a nice diagram that was in our slides last year. Um, it's the same thing again. Arginine combines with glycine to form this intermediate, and then this intermediate combines with a methyl group from SAM to give us creatine. That's the endogenous source of creatine. And then you probably heard of people taking creatine supplements, like bodybuilders and stuff. It's the exogenous source of creatine. And then creatine enters your muscles through a creatine transporter. And when it's in the muscles, it can be converted into phosphocreatine or back to creatine. It just keeps going back and forth. Right? And this production of creatine is done in the liver and the kidneys. Okay, so remember it's done in the liver and the kidneys, and then it goes to the muscles. In the muscles, creatine can be broken down into creatinine. So creatinine is like the waste product of creatine. After it's metabolized and broken down, it gives us creatinine. And then it goes to the kidneys to be excreted. So when you say creatinine, that's the waste product, and creatine is the normal one that we use. Okay. So creatine metabolism, creatine is broken down to give us creatinine, and then it's transported out of the cells to the blood. So it goes out of the muscle cells or wherever it's stored, and it goes to the kidneys, and the kidneys will clear the creatinine or filter the creatinine out of the blood to excrete it. Okay, and the breaking down creatine into creatinine, uh, this process we call it cyclization. So cyclization is basically when you form a ring. So normally creatine is like a linear structure, but cyclization turns it into a ring structure. Okay. So we said that creatinine is cleared by the kidneys. So this is actually a way that you can determine or measure how well the kidney is functioning. So normally it's supposed to clear it from the blood. You're not supposed to have high levels of creatinine in your blood. So if you see that you have a high serum creatinine, that means that your kidneys are not working well enough. They're not filtering the blood. So creatinine will be high and the kidney has some sort of damage. Okay, so let's try to recap what we just said. Glycine, remember glycine is important for what component of red blood cells? Heme. And heme gets the carbon and nitrogen from which two precursors? Remember guanosine, sucking blood, glycine, and succinyl-CoA. And this is the porphyrin ring that has the iron in the middle. So this whole structure, we call it heme. Like synthesis of creatine. Creatine is synthesized where? In the liver and the kidneys. From which three components? Remember creatine, the mnemonic creatine gives ATP to the muscles. So GAM, glycine, arginine, and SAM, methyl group from SAM. And remember how I mentioned the cyclization? This is what it looks like. So over here, it's like linear, and then here it looks more like a ring. So that's the cyclization. And the creatinine is what is going to be excreted. Like now for the products of arginine, we already mentioned creatine now. So next is urea. So what is urea? First of all, normally when we are metabolizing proteins, when we're breaking down proteins, we get this really toxic metabolite that we don't want in our body. So we don't want ammonia. We want to get rid of it or make it less toxic. So what our body does is that it detoxifies ammonia. It makes it a more safe version, and we call that urea. So we convert ammonia into urea through the urea cycle. And what gives rise to urea is arginine. So the amino acid arginine is what gives us the urea, which is what we're going to excrete in the urine. And urea is the less toxic version of ammonia. So that's for urea from arginine. The last thing that arginine gives us is nitric oxide. So in general, the function of nitric oxide is, it's not just in the blood vessels, it's also in the brain and in other areas as well. This, for the main function in the blood vessels, it helps in vasodilation. So it's actually found in the endothelial layer here. It's synthesized here and then it travels to the smooth muscle cells around it. When it goes to the smooth muscle cells, the cells relax, and this causes the blood vessel to dilate and widen. Type, so it causes vasodilation. And we get this nitric oxide from the amino acid arginine. So arginine combines with NADPH with oxygen, and through the enzyme nitric oxide synthase, again, the enzyme makes sense, synthase because it's synthesizing nitric oxide. And this just shows you that you have like more than one version of nitric oxide synthase. There's one in the endothelium, one in the nerves, the neuronal nitric oxide synthase. So I don't think you guys need to know this. Okay, so again, arginine gives us three things. Remember, what was the thing that it gives us in the muscles? Creatine. And what's the other product? Urea. And then finally, we got last one we just mentioned, we get nitric oxide. Okay, now for the I think this is the final amino acid. For glutamate, we get from glutamate we get GABA. Like, what is glutamate and what's GABA? These are two neurotransmitters that are found in our brain that have opposite functions. So one of them is made for excitation, and one of them is made for inhibition. So to remember that, glutamate, remember excitate. So excitate is not actually a word, but just because it sounds similar, so glutamate, excitate, remember that it causes depolarization. So the sodium ions will enter, and you will have depolarization, so more action potential. GABA, on the other hand, it will be inhibitory because it causes more chloride to enter. So the charge will become so negative. We call this hyperpolarization, and so no action potential can occur, and this is going to cause inhibition. Okay, so glutamate, excitate, GABA, inhibit. And the way that glutamate gives us GABA is by an enzyme known as glutamate decarboxylase. Okay, so glutamate decarboxylase also needs vitamin B6. Like, and again, you could look at it, it makes sense. You'll see that the carboxyl group, COOH, is going to be lost, and you're going to form GABA. So the enzyme, what is the enzyme called? Glutamate decarboxylase. It removes a carboxyl group. And what cofactor does it need? Vitamin B6. Okay. طيب. Now we have something known as branched chain amino acids. First of all, what are they? So branched chain amino acids are amino acids that have carbon atoms in chains or in branched chains. What does that mean? So when you look at a normal amino acid, you can draw one line, like one continuous line. So you could, if you look at the carbons, they're connected in just one pathway, even if it doesn't look like a line, it's just one. On the other hand, for the branched chain amino acids, you're going to have chains coming out. I can't just draw it in one go. Right, so you can see here branches. So we call them branched chain amino acids because of that reason, and they're all essential, meaning that you have to obtain them from your diet. You need to get them from your food. But now that we know what they are, how are they metabolized? How are they broken down in our body? We have three main steps. There's actually more steps, but the doctor focused only on these three. We're just gonna look at these. The first one is transamination, then decarboxylation, and then dehydrogenation. So let's start with transamination. From the name, it also makes sense. You're moving an amino group somewhere else. You're removing an amino group. So that's transamination. What do you produce after this transamination? You produce an alpha keto acid. So an alpha keto acid just means that you have a ketone group in the alpha carbon. So the alpha carbon is just like the central carbon, and the ketone group means you have C double bond O. Okay, so transamination gives us an alpha keto acid. And then this alpha keto acid will undergo decarboxylation. So decarboxylation is again the removal of carbon dioxide or a carboxyl group. And this is done by this enzyme. So this enzyme is really important. We're going to go back to it again. It's called the branched chain keto acid dehydrogenase. Or they'll also say alpha keto acid dehydrogenase. Type. The third step is dehydrogenation, where you remove a hydrogen. Okay. So I like this diagram the doctor put it, it's more clear. The first step was transamination by the transaminase enzyme, and then decarboxylation by the branched chain keto acid dehydrogenase. Okay, and then we have the dehydrogenase type. So this one produces alpha keto acids. Remember that the product here is alpha keto acid. And you also need to know the products from each one. So you need to know which one gives you succinyl A, which one gives you both succinyl A and acetyl A, and so on. Okay, yeah. So for the enzyme, branched chain alpha keto acid dehydrogenase, this enzyme needs 5 cofactors to function. So these five, I tried to come up with a mnemonic that has to do with branch. So when you think of branch, you think of a tree. So branches of a tree. And when you think of trees, you can remember trees' leaves change in fall by nature. I know that's also not the best mnemonic, but if it works, hopefully it helps you guys remember. So what does each one stand for? The T is for thiamine pyrophosphate, L is for lipoic acid, C is for coenzyme A, and F is for FAD, N is for NAD. You need to remember each of these cofactors also comes from a vitamin, except for lipoic acid. But thiamine pyrophosphate, you need to know it comes from B1, vitamin B1. Coenzyme A comes from B5, FAD comes from B2, and NAD comes from B3. So remember what vitamin gives rise to each cofactor. Right. So we talked about how the branched chain amino acids are broken down, but sometimes in some people, they might have a deficiency in this really important enzyme known as the branched chain keto acid dehydrogenase. Like what happens when you have a deficiency here? Whenever you have any deficiency, like as a rule, when this is not working, everything before it would rise, right? So it makes sense because you're not breaking down the ones before it, so they're just going to increase, and everything after it will decrease. So because this is not working, if you have a deficiency, you'll have a rise in alpha keto acids and a rise in the branched chain amino acids. So high valine, high isoleucine, high leucine, and then high alpha keto acids of each one. Okay, so when is this the case? When do people have a problem in the branched chain keto acid dehydrogenase enzyme? In a disease known as maple syrup urine disease. And the reason they call it that is just because the urine smells sweet, so maple syrup urine. Okay, so let's talk about the cause, presentation, diagnosis, and treatment. For the cause, again, we mentioned that it's a deficiency in the enzyme here, the branched chain keto acid dehydrogenase. For the presentation, you're going to see neurotoxicity. This is because leucine is going to be damaging to the structures in your brain and the nerves. And the second thing is sweet smelling urine. We already mentioned that the urine smells sweet. That's why the disease is called maple syrup urine. And this is caused by the isoleucine keto acid. So remember what causes what. So remember which one is the one that causes the smell and what causes neurotoxicity. And then we also have lethargy, which means you feel very tired, irritability, apnea, which is like a difficulty in breathing, and seizures. But this occurs in the first few days of life, meaning that the baby or the infant will show these symptoms immediately. And the reason for that is because when they're taking the milk, milk has branched chain amino acids in it. So right away when they start breastfeeding, they're going to present with the symptoms. It's not going to take time. Okay. Now for diagnosis, again we said we have high valine and high keto acids. So you'll see high levels of not just valine, sorry, high valine, isoleucine, and leucine. So high branched chain amino acids and high alpha keto acids. And for treatment, obviously you're going to restrict the branched chain amino acids because they're just accumulating and causing these toxic metabolites to accumulate. They don't want that. They just restrict the branched chain amino acids from their diet. Another thing is you supplement thiamine. So remember, thiamine was one of the vitamins here. Thiamine is vitamin B1. So if you supplement B1, they're gonna have more thiamine pyrophosphate, and it's one of the cofactors that is going to help support this enzyme. And lastly, just monitor plasma amino acid concentrations because you need to make sure that they're not having like the disease is not progressing while you're not noticing. Because if it progresses and you don't notice or you don't diagnose it, it will cause really damaging effects to their nervous system, like mental retardation, seizures, and so on. You need to catch it early on, and it might even be fatal if you don't catch it early enough. So just keep monitoring plasma amino acid concentrations, supplementing thiamine, and restrict their diet from the branched chain amino acids. So let's test ourselves again. What is the enzyme that is really important? Branched chain keto acid dehydrogenase. What are the cofactors? So what's the cofactor we get from B1? Thiamine pyrophosphate. What do we get from B5? Coenzyme A. And from B2, FAD. And then from B3, NAD. Okay. So what is the substance, or what is the keto acid? The keto acid of which amino acid gives the sweet smell? It's isoleucine. And you can remember this because of sweet and smell SS, and there's also an S here, so it kind of might help. And what is the main problem in maple syrup urine disease? It's neurotoxicity. And again, it's mainly due to accumulation of which amino acid? Leucine. Okay. And one of the ways you could treat is by supplementing what? Which vitamin? B1 or thiamine. Okay. So now we're done with the branched chain amino acids. Now we're going to talk about methionine, cysteine, and homocysteine. We have these three amino acids. Let's start with methionine. Methionine is an essential standard amino acid. Remember, we said essential meaning you have to get it from your diet. So you don't produce it in your body. You need to get it from the diet. Cysteine, on the other hand, is non-essential. So you don't need to get it from the diet. But I need to mention something here. It's only non-essential if you have methionine, because cysteine comes from methionine. So if someone has no methionine, deficiency in methionine, you have to supplement cysteine, and it becomes essential in that case only. So it's only non-essential when you have methionine. Okay, and then we have homocysteine. Now, homocysteine is a non-standard amino acid. I've been saying standard. What does standard mean? How does it differ from a non-standard amino acid? When we say non-standard, that means it does not make up proteins. And normally when you take a protein and you break it apart, it's made up of amino acids, right? These amino acids, we call them the standard amino acids. Non-standard amino acids, they are not they're not the building blocks of proteins. But the reason we still call them amino acids is because they have a similar structure to the standard amino acids. So they're still made up of an amino group and central carbon, etc. Okay, so methionine actually gives us homocysteine, and then that gives us cysteine. So if we don't have methionine, what happens? Cysteine becomes essential. Let's talk about the pathway. How do we get methionine and cysteine and so on? So first, I'm going to start simple, and then I'm going to add on the cofactors and the enzymes and so on. For methionine, you add an adenosine group from ATP to form S-adenosyl methionine. Okay, so methionine, adenosine group, S-adenosyl methionine. Remember, that's our methyl donor. And then it removes a methyl group, it becomes S-adenosyl homocysteine. And then this removes an adenosine group, it becomes homocysteine. Now that we have homocysteine, it has two options. It will either go through the remethylation pathway or through the transsulfuration pathway. So if it goes through the remethylation pathway, you just add another methyl group and you go back to methionine. The other option is the transsulfuration pathway, where it will add a sulfur group and it becomes cystathione. And then cystathione will give us cysteine. Now, cysteine is really important. I'm not sure if you guys took the free radical lecture yet. I don't remember where, if it was MOL 1 or MOL 2. But basically, we have this really damaging substances in our body known as free radicals that the way we fight them is by antioxidants, and cysteine is very important in fighting these free radicals. So it is an antioxidant. Okay, cysteine is very important for protecting your body from these reactive oxidative species or free radicals. Okay, so now let's go into more details in this pathway. We said methionine, you add an adenosine group, it becomes S-adenosyl methionine. Now SAM will give up its methyl group, it becomes S-adenosyl homocysteine. Now when it gives up its adenosine group, it becomes homocysteine. Now we have two options. So let's start with the remethylation pathway. So let's start from up here. Folate, which is vitamin B9, is going to convert into 5-methyl tetrahydrofolate. With this enzyme here, methylene tetrahydrofolate reductase, or MTHFR. Okay, so folate to methyl tetrahydrofolate. We just care about the methyl here, we just want this part. Now, it will give the methyl group, so we'll be left with tetrahydrofolate, and the methyl group combines with homocysteine to give us methionine. Okay, so we take the methyl group, give it to homocysteine, it becomes methionine. And this is done with the help of the enzyme methionine synthase. Again, the enzyme makes sense, synthase, because you are synthesizing methionine. And it needs vitamin B12. So here you have vitamin B9, and here you have vitamin B12. 12. This is vitamin B9, and this one just needs a cofactor B12. Right, so that's for the remethylation pathway. The other pathway is the is the transsulfuration pathway. What happens is homocysteine will combine with serine to give us cystathione, and this is done with the enzyme cystathione synthase. Okay, so again, it makes sense. You are synthesizing cystathione, so cystathione synthase, and it needs the cofactor B6, right? And then cystathione will be converted into cysteine, and these are just like some other byproducts. I don't think they're very important. But basically, you have alpha ketobutyrate and succinyl-CoA. I think just know them just in case. So that's it. But now, what is homocystinuria? So sometimes you could find patients with high levels of homocysteine in their blood and in their urine, and this is not good because homocysteine actually has very toxic effects. Okay, so we don't want to have homocysteine elevated. But what causes this elevation? If it's a mild or moderate elevation, then it could be like a deficiency in vitamins. Remember we said here we need vitamin B9, here B12, and here B6. So it could be either of these three, a B6 deficiency, B9, or B12. Right? What if it's a severe homocystinuria, like a very high level of homocysteine? Then this is probably a defect in the enzymes, not the cofactors. And that would be caused by some disorder that's genetic. So it's normally autosomal recessive. So you have to receive a gene from your dad and from your mom. So it's from both sides. Okay, so what are the enzymes that could be defective? Most likely it's going to be cystathione synthase. Okay, so cystathione synthase is the one that's in classic homocystinuria. And another like possibility is you could have methionine synthase deficiency or MTHFR. So it could either be a deficiency here, or here, or here. And in all of these cases, if these are deficient, can you guess what's going to accumulate? It's this one, homocysteine, because you're not going down, you're just going up. So you're just going to keep accumulating homocysteine. Whenever you take in, like you eat anything that has, is going to result in more methionine in your blood, it's going to be broken down to homocysteine. But what is like the level in which you know it's mild or moderate or severe? First of all, normally it's supposed to be 5 to 4. If it rises to 20 to 100, 20 to 100, it's mild to moderate. If it's more than 100, then you call it severe. Okay? So that's how to diagnose. Now for the presentation, again, we said that homocysteine is very toxic. So what are the effects? What are the toxic effects of homocysteine? So first, it affects your brain. It causes mental retardation and problems in your cognitive function. Another thing is that it affects your lens. So in your eyes, you have connective tissue that holds your lens in place. So homocysteine will come and damage this connective tissue, so your lens will not be held in place and it will move, and this will cause like vision problems. And then will also affect your collagen. And you know that collagen is very important for building a good framework for your bones to be growing on, and it will also affect how your skeletal system is built. So you'll find that you have long limbs and chest deformities. Finally, it's going to cause atherosclerosis and blood clots. It has damaging effects on the endothelium. It causes inflammation. More immune cells come, and when you have inflammation, immune cells in general, they start to like release or spit out these damaging substances that cause plaque. So when you have plaque forming in the blood vessels, it causes narrowing and hardening. We call that atherosclerosis. And the blood clots are going to lead you to be more susceptible to having a stroke or a heart attack. Okay, so that's for the presentation. Remember, mental retardation, lens dislocation, long limbs and chest deformities, and then atherosclerosis and blood clots. Now for the treatment, how do we treat classic homocystinuria? Number one is you avoid methionine. You don't want to have methionine because if you ingest it, what's going to happen if you have like food that has methionine, you're going to just have accumulation of homocysteine, and that's just going to have toxic effects. We don't want that. Avoid methionine. Number two is increase cysteine. So remember how we said if you don't have methionine, cysteine becomes essential. So now that it's essential, you need to make sure that it's included in your dietary intake. And finally, supplement B6. So this is because in classic homocystinuria, the enzyme that is defective is this enzyme. and the cofactor for it is B6. So some people are found to be responders of B6, meaning that if you give them B6, they might possibly improve, or the symptoms will improve. So let's try to test ourselves. Methionine is essential. What about cysteine? It's not essential. And homocysteine, it's non-standard. And finally, if we go through the transsulfuration pathway, what will homocysteine become? Will it go back to methionine, or will it turn into something else? It will turn into something else, which is cysteine. Okay, so the first step, we said that methionine takes an adenosine group from ATP. What does it form? SAM, or our methyl donor, acetylcysteine methionine. طيب, SAM donates two things to form homocysteine. What are they? A methyl group and an adenosine group. And finally, homocysteine combines with serine to give us cysteine. So what is the enzyme and cofactor that helps in that? The enzyme is cystathione synthase, which needs B6. And you can kind of remember it because cystathione sounds like sitta. So 6 in Arabic is sitta. So sitta, cystathione. Okay, cysta needs sitta. This is like a recap of the whole pathway. Homocysteine to give us methionine. What is the enzyme? Methionine synthase. طيب, yeah. For methyl tetrahydrofolate to form tetrahydrofolate, what cofactor does it need? B12. And finally, what gives us this methyl tetrahydrofolate? Folate or B9. Okay, so here's B9, here's B6, and B12. طيب, classic homocystinuria is deficiency in which enzyme? Cystathione beta synthase. And what can you supplement in some patients to improve homocystinuria? B6. A few causes that are less common are deficiencies in the two other enzymes, methionine synthase and MTHFR. Right? And finally, what is the enzyme that turns folate into methyl tetrahydrofolate? Remember the long MTHFR. Okay, so this is just a summary if you guys need to look at it to remember which enzyme needs which cofactor. And that's basically it. I hope everything was clear. If you have any questions at all, you can contact me at any time. And good luck in all your exams. And Ramadan Mubarak. Thank you.